Running an optimization under genetic algorithm. 96. Click on Settings. Go to the Engine tab and change the optimization mode to manual, then optimize using genetic algorithm. 98. Insert the value 78 on cells C2:F2 and proceed to start over another optimization, this time with GA. 99. Go to the Diversity tab during the simulation run. With the GA engine operating, the Diversity tab is shown. The plot on the Diversity tab assigns colors to adjustable cell values, based on how much the value of a given cell differs across the population of solutions that are stored in memory at a given point. Here, you will see 4 columns for each one of the solutions proposed for each metal. Using GA terminology, this is an indication of the diversity that exists in the gene pool. Each vertical bar in the plot corresponds to one adjustable cell. Horizontal stripes within each bar represent the values of that adjustable cell in different organisms, or multiple solutions. The colors of the stripes are assigned by dividing the range between the minimum and maximum value for a given adjustable cell into 16 equal length intervals. Each of the intervals is represented by a different color. For example, if a vertical bar is single color, the corresponding adjustable cell has the same value for each solution in memory. The diversity diagram can give us valuable cues with respect to ending the optimization. When all columns become the same color, maybe it is time to stop the optimization process since the GA has found an answer for which all possible subsolutions generate the same, therefore, homogeneous value. There is, however, no definite proof that we have achieved an absolute maximum or minimum. The population size tells the optimizer how many organisms, or complete sets of variables, to store in memory at any given time. With still much debate and research regarding optimal size to use on different problems, we generally recommend using between 30 to 100 organisms depending on the size of your problem, bigger population for larger problems. A larger population takes longer to settle on a solution, but is more likely to find a global solution because of its more diverse gene pool. One difficulty with optimization, when your problem has seemingly endless possibilities, is in determining where to focus your energy. In other words, how much computational time should be devoted to looking in new areas of the solution space and how much time should be devoted to fine-tuning the solutions in the population that have already proven to be fairly good. A big part of the GA's success has been attributed to its ability to achieve this balance. The structure of the GA allows good solutions to breed, but it also keeps less fit organisms in memory to maintain diversity, reasoning that a latent gene will prove important to the ultimate solution. Crossover and mutation are two parameters that affect the scope of the search, and RiskOptimizer allows you to change these parameters before, and also during, the evolutionary process. This way, you can help the GA by deciding where it should focus its energy. For most purposes, you should not need to adjust the default crossover and mutation settings. However, if you want to fine-tune the algorithm to your problem, conduct comparative studies, or just to experiment, you can try adjusting them. The crossover rate can be set between 0.01 and 1.0, and reflects the likelihood that future scenarios or organisms will contain a mix of information from the previous generation of parent organisms. A rate of 0.5 means that an offspring organism will contain roughly 50% of its variable values from one parent and 50% from the other parent. A rate of 0.9 means that roughly 90% of an offspring organism's values will come from the first parent and 10% will come from the second parent. A crossover rate of 1 means that no crossover will occur, so only clones of the parents are evaluated. The default rate used by RiskOptimizer is 0.5. Once it has started solving a problem, you can change the crossover rate. The mutation rate can be set to between 0.0 and 1.0, and reflects the likelihood that future scenarios will contain some random values. A higher mutation rate means that more mutations, or random gene values, will be introduced into the population. Because mutation occurs after crossover, setting the mutation rate to 100% random values, will effectively prevent the crossover from having any effect, and RiskOptimizer will generate totally random scenarios. If all the data of the optimal solution is somewhere in the population, then the crossover operator alone is enough to eventually piece together the solution. Mutation has proven to be a powerful force in the biological world for many of the same reasons that it is needed in a genetic algorithm. It is vital to maintaining a diverse population of individual organisms, thereby preventing the population from becoming too rigid, and unable to adapt to a dynamic environment. As in a genetic algorithm, it is often the genetic mutations in animals that eventually lead to the development of critical new functions. Usually, the default mutation setting does not need adjustment. However, You can change it to fine-tune performance on complex problems. You might want to boost the mutation rate if the population is fairly homogeneous and no new solutions have been found in the last several hundred trials. Typical setting changes are from 0.06 to 0.2. Once RiskOptimizer has started solving a problem, you can change the mutation rate dynamically. The auto setting ensures auto mutation rate adjustment is selected. This adjustment allows RiskOptimizer to increase the mutation rate automatically when an organism ages significantly, that is, when it has not changed over an extended number of trials. For many models, especially where the best mutation rate is not known, the auto setting is preferred. For example, we ran this optimization process using GA and the proposed portfolio gave us the following answer. This portfolio is substantially different from the first one we generated with automatic method. On the next section, we will devise a functionality that will allow the comparison of both methods.